All right, y'all, here we go with our first of two videos where we will focus in on the ATP synthase molecular machine that helps us create or synthesize clearly the ATP. All right, when we look at our metabolism map, we are still talking about energy metabolism. We took those high energy electrons down the electron transport chain where we made water. And so remember, there we were right down here. Okay, and the energy harnessed from the creation of that proton gradient is what is going to be used to drive this ATP synthase molecular machine to synthesize these ATP molecules. All right, so let's kind of review first the citric acid cycle. Let me grab that pointer. So the citric acid cycle right here oxidize the acetyl fragment of acetyl-CoA to give us that carbon dioxide. And in the process of that oxidation, we captured a lot of high energy electrons in the form of NADH, okay, which we see, and FADH2. And the key function of that citric acid cycle was to harvest those high energy electrons from the carbon fuels. And then this is coupled, so today we're going to be talking about coupling the electron transport chain, which was all of these first four complexes, to our ATP synthase, that complex 5 in our cartoon. So just as a review, complex 1 got electrons from the citric acid cycle, so we saw that NADH feed in here. And we also get some from glycolysis and other places. So we get NADH from multiple sources going into complex 1. Electrons from that citric acid cycle went directly into complex 2 through, remember that, FADH2, which passes them off to the coenzyme Q over there at complex 2. And so the electrons in 1 and 2 are picked up by this coenzyme Q and carried into complex 3, okay, where they're passed to cytochrome C, which passes them off into complex 4, where that oxygen is reduced to water. We saw that complex 1, 3, and 4 are all protein pumps. So remember, over here we are in the inner membrane space. Up here we're in the matrix, so our outer membrane would be somewhere around here. And the goal was to set up a proton gradient with more protons out here than in the center. All right, so we've been pumping the protons out in each of these three complexes. And that's where we are, and now we're going to get to the ATP synthase. All right, we have two pictures here of our ATP synthase. So ATP synthase is ubiquitous. It is in all living things. Despite that, we've never isolated all of the subunits together where we could do X-ray crystallography or electron microscopy on it. Instead, what we have is we've isolated different parts from a variety of organisms to study them, and this picture is kind of a compilation of our protein modeling, piecing together the best model we have. Okay, over here on the right, we have our cartoon drawing, which simplifies it a little bit. This is, even though we are still actively working on this, it is a very solid molecular modeling. We've had lots of people studying it for a long period of time. Okay, so to look at this, we need a common terminology. So first of all, let's orient ourselves. So we have up here the matrix, and we have down here the intermembrane space. All right, I'm going to pause for a minute to kick the cat out, or at least let her into the windowsill so she will quit walking on the screen. There we go. All right. We have multiple subunits that are broken down into two sections. So we have this F0 section, okay? That is the transmembrane portion. Okay, it is made out of subunits A, B2, by which we really mean there are two B subunits, and C. And each of these cylinders here, shown as cylinders, okay, are a C subunit. So there'll be many of those. And then, whoops, that's not what I wanted. In the F1 section, we're going to have alpha subunits, okay, shown in orange over there, beta subunits, shown in yellow, gamma shown in pink, epsilon shown in green, and the delta, so I went out of order here, but you got delta shown down here in this purple piece right there. So notice that we use the Greek letters for the F1 subunits. We use A, B, and C for the F0 pieces. All right, so now let's look at the role of each of those subunits. All right, so first, even though it's written down there at the far bottom right, we're going to look at what we call the knob, and that is the bottom piece. So the alpha and beta subunits, so right in here. Okay, the knob is made of 3 alpha and 3 beta subunits. They're tightly associated as alpha beta dimers. Okay, so each alpha is tightly associated with a beta. We'll sometimes call this, instead of the knob, we could also call it the alpha beta hexamer. Okay, because there's 6 subunits, 3 alpha, 3 beta. And the role, this is our catalytic portion. This is where our active site is. The active site is actually on the beta subunit. Okay, so this is where that activity of ADP reacting with the phosphate, forming ATP occurs on these beta subunits. All right, so that was the knob. So then we're going to talk about what we're going to call the rotor, which can also be called the central stalk or the axle. Okay, and this is the gamma and the epsilon subunits. Okay, and they are tightly associated with each other. So we're looking right here, the pink and the green. Okay, and this is the part that turns. This spins. And it spins 360 degrees, spins all the way around. Okay, and this comes down, notice, into the center. So these alpha and beta, the knob forms kind of a barrel shape, and that gamma piece comes down into the center. So it's spinning, and it is closely associated with these alpha and beta pieces. The alpha and beta, this subsection of the knob, stays stationary, but this axle piece spins within there. All right, so going over to the left, we have the peripheral stalk. So the stuff, not all the stuff in purple, actually, well, a little bit. We've got the A subunit. I guess it is all the stuff in purple. The B2, the 2 B subunits. And we have that delta subunit. And these pieces serve to stabilize the knob. Okay, so they stabilize that hexamer. So that it doesn't spin. They hold it in place so it doesn't spin. Okay, and since stabilizing, that's where this gets the name stator, sometimes. Peripheral stock is what I'll generally use. Not mentioned as a part of that, but just to know that this alpha, or I'm sorry, not alpha, this A subunit right here is also the pathway where the protons will enter and exit as they're passing across the gradient. So remember, we've built up more protons out here, higher concentration than here. So the protons are going to move, and this is where they will enter and exit. And we're going to look at that in just a minute. All right, and then the C ring up here. So all of these blue, shown as kind of blue cylinders right here. Okay, we will have anywhere from 8 to 15 C subunits in that ring. Okay, invertebrates, we have 8. They're all tightly associated with each other, and the ring spins 360 degrees. Okay, as we talk about this as a molecular motor, it really is what we think of as a motor. It is really, it's moving and causing things to change. Okay, this ring right here is tightly associated with the stalk, actually with both of them. Okay, but as we think about it with this central stalk or this rotor, so as the C ring spins, it causes this to spin. But the C ring spins faster, and the peripheral, or I'm sorry, not the peripheral, the central stalk kind of builds up. As the C ring spins, it builds up tension, builds up tension, builds up tension, and then finally this will kind of jump. And so the central stalk spins in kind of 120-degree jolts. So instead of a smooth spinning motion, it's going kind of jump, jump, jump all the way around. Okay? All right, so that's the parts, the structure, the functionality. And so what we want to talk about now is, what does that rotor spinning have to do with the activity of the active site? And how does the A subunit and the C ring allow the protons to go through and cause this C ring to spin? And before we look at that, we want to look just a little bit at the kind of structure of this section. So let's orient ourselves. So we have, here we're showing the outer membrane. Okay, so over here, we're in the matrix. We've got our inner membrane, and not labeled, but it should be, right? This is our intermembrane space. All through here, between the outer and the inner membrane. So when I look over on the right at my mitochondria, notice that the yellow is showing the outer membrane. The blue is showing that inner membrane. And remember how we have those folds, these cristae, that this all folds in and out of itself, right? The inner membrane space, or I'm sorry, not the inner membrane space, the matrix is all in here. The inner membrane space is between the yellow and the blue. What we want to look at right now is all of these folds, okay? Because it turns out that we have dimerization. See if I can spell that. Dimerization of the ATP synthase. Okay, you can see that here, where they're grouped together. And that grouping together causes this inner membrane to fold or pucker. Okay, so that's what's causing all of these folds right here. And if we look at why that's important, all right, what is shown in yellow here, all right, is our electron transport chain. So we know our electron transport chain is helping us set up that proton gradient. And all of these little pink circles are showing protons, right? So we're pumping the protons into that inner membrane space. And what we need is a proton gradient. We need to have a different concentration of protons in the inner membrane space than we have in the matrix. But we don't have to have it totally different across the entire intermembrane space. We just really need a locally high concentration. And so having these folds and these puckers where the electron transport chain is closely associated nearby the ATP synthase helps us more quickly and easily set up this proton gradient. Okay, because we pump those protons in here, and then because they're kind of in this little pocket, we more quickly set up this area of high concentration. All right, so back to our question. So if you remember, I was saying that what we really now want to talk about is why does that that rotor spinning cause the active site to work, and how do the A and C subunits allow the protons to go through? Okay, so let's orient ourselves here. We're going to talk about what we call the binding change mechanism. Okay, it accounts for the synthesis of ATP in response to the proton flow. And so what we're going to see is that the spinning action causes a conformation change, and this binding chain mechanism describes for us how that active site works. All right, so first though, we need to orient ourselves. So when we look at this picture, this is what I'm looking at. All right, I am looking at that knob, and I'm looking at the rotor, specifically that gamma portion, that one shown in pink of the rotor. So you can kind of picture looking up from the bottom, and I would see 3 subunits, or 3 of these alpha beta dimers, right? So we've got alpha beta dimers, 3 of them. So one shown in purple, one shown in blue, one shown in green. Don't worry yet about why they have different letters. We're going to look at that very next thing. Okay, the triangle here is our gamma kind of rotor that's spinning. And what's kind of important is that this thing is not perfectly symmetrical. So each side of that unit has different conformations. So you can picture it as like, you know, a different shape kind of the 2 sides, and then the pointy end piece here. So each side looks different, and so it's going to affect the conformation of these alpha beta dimers differently depending which side of this gamma they are associated with.